Today, I'm going to talk about iron homeostasis and the immune system. Iron is an essential element for life. It plays a key role in energy production, DNA synthesis, and cellular metabolism. In this presentation, I will explore how iron is closely linked to the immune system. It influences both protective responses and disease outcomes. A key concept is the competition for iron between the host and pathogens. This competition strongly affects how infection develop and progress. I will also discuss how iron imbalance impacts immune function, how deficiency and overload can disrupt normal immune responses. In addition, I will examine emerging therapeutic opportunities related to iron regulation. Finally, I will highlight important research gaps in current research. Let's begin by understanding why iron is so biologically important. Iron is vital because it can switch between different oxidation states. This ability allows it to participate in redox reactions that are fundamental for cellular metabolism and energy production. It acts as a critical cofactor in many enzymes, especially those involved in mitochondrial respiration, DNA synthesis, and DNA repair. Without iron, these essential biological processes cannot function properly. Because of its role in energy production and biosynthesis, every cell type in the body, including immune cells, require iron for survival, growth, and proper functioning. Although essential, iron can be harmful in excess. Too much iron promotes the formation of reactive oxygen species, which can damage cells. Therefore, strict regulation is crucial to maintain balance and protect biological systems. Given its importance, iron becomes a central point of competition during infection. During infection, the host actively limits the availability of iron to invading pathogens. This defense strategy is called nutritional immunity and is designed to restrict microbial growth by depriving them of an essential nutrient. The host achieves this by tightly binding iron within proteins such as transferrin and ferritin, as well as storing it inside cells. This reduces free iron levels in circulation, making it less accessible to pathogens. In response, pathogens develop strategies to overcome iron restriction. They produce molecules called siderophores that scavenge iron or express specialized receptors that help them extract iron from host proteins. This ongoing competition creates an evolutionary arms race between host and pathogen. Ultimately, the ability of either side to control iron availability can determine whether the infection is cleared or progresses. Beyond this competition, iron also directly regulates immune cell behavior. Immune cells, particularly T lymphocytes, require substantial metabolic energy when they become activated and begin proliferating. This supports their rapid growth and functional responses during immune activation. Iron plays a key role in enabling mitochondrial energy production and biosynthetic pathways. These processes are essential for supporting the increased metabolic demands of activated immune cells. When T cells are activated, they upregulate iron uptake mechanisms, especially through transferrin receptor pathways, to ensure they can access enough iron for their functional needs. If iron is limited, immune responses are weakened. leading to reduced proliferation and altered effector functions. In contrast, adequate iron levels support strong and effective immune activity, making iron a key regulator of immune performance. However, maintaining the right level of iron is critical, as an imbalance can be harmful. Both iron deficiency and iron overload disrupt the normal balance of immune system function, known as immune homeostasis. This balance is essential for proper immune regulation and response. Iron deficiency is common globally and leads to weakened immune responses. As a result, individuals become more susceptible to infections due to reduced immune effectiveness. In contrast, excess iron can increase oxidative stress and inflammation. It may also support pathogen survival and contribute to tumor growth, creating additional health risks. The immune system is highly sensitive to iron levels, so tight regulation is crucial. Disruption of this balance contributes to infections, inflammatory disorders, and even cancer development. This understanding opens the door to therapeutic strategies targeting iron metabolism. Targeting iron metabolism offers important opportunities in medicine because iron is central to both immune function and pathogen survival. It can be exploited for therapeutic benefit. Lowering iron availability can help control infections by restricting the growth of pathogens that depend on iron for survival and replication. Restoring iron levels in deficient individuals can enhance immune function. In cancer, manipulating iron metabolism may strengthen immune responses or directly affect tumor cell survival. Because iron is involved in many essential biological processes, any therapeutic intervention must be carefully controlled. Incorrect modulation can lead to unintended and potentially harmful effects. Despite these advances, several key questions remain unresolved. We still lack a detailed understanding of how different immune cell types sense and regulate iron at the molecular level. This is a key gap in current immunology and iron biology research. The interaction between whole body iron homeostasis and local tissue specific immune responses remains poorly defined. Understanding this connection is important for linking physiology with immune function. The long term consequences of altering iron levels, especially in chronic diseases, are not yet well understood and need more investigation to ensure safety and effectiveness. A major challenge remains in developing therapies that can precisely and safely target iron metabolism without causing unintended effects in other biological systems. This highlights the importance of continued research into iron biology as a central regulator of immunity. Thank you very much for listening to this presentation.